All right. Well, let's get started. Welcome to immunology. So, okay. So today I'm just going to start with an overview of both how we set up the course here, and then the second half is going to be a quick overview of the immune system just to get your feet wet. Okay. So it's always about 15 minutes. 15 minutes is often shorter than that. Almost always Monday at 10 in this conference room. If you miss it, we try and put everything on website, iTunes, YouTube, but then this year I started website 312immunology.org, where I'm going to post them as well. Not last year. I couldn't find them last year, but 2 years ago, the Abbas series is posted on there if you want to rewatch. I'm also going to put all the Jeopardies on there as we go through them that you can download and just use as a memorization tool if you want. Okay, so the lectures are in a format where I kind of outline the reading. It most certainly will not replace the reading. Go through it once before the lecture. And then after we've done a series of about 3 or 4 chapters, then we try and take a pause and do a Jeopardy, just kind of a quiz review of some of the things that I think are worth memorizing. That does not mean anyone else in the world thinks they're worth memorizing. It's just best guesses. Me, the things that you should probably start memorizing. All of this is aimed at being prepared for the boards. There are going to be interruptions in the schedule from time to time for things like the college meeting, holidays. I tend to not do anything over the holidays. During those times, especially since we're starting January this year, which is going to be new, it's very dense, so don't forget to read some during that week, too. I'd be probably reading all year long, some part. Okay, so we're going to start with by going over the book How the Immune System Works by Lawrence and Pyrak. I love this book. I think it gives a really great, straightforward overview and all the high-yield facts. I think the worst part about immunology is Dr. Belanti always says it's trying to see the forest through the trees. So we pick up all these little pieces, but being able to put it together in one big picture, I think, is sometimes challenging. And then this year, instead of Abbas, we're going to go over Janeway's Immunobiology, 8th edition. So we'll see how this goes. We're going to be assisted by a group in New York who's going to help come up with some questions, who also uses this course. So I'm looking forward to that collaboration. And I don't know how we're going to do it year to year. We'll see how this book is, and then I just today got a copy of the latest edition of Abbas. The last edition wasn't great. So it may be that we go back and forth, and then every year you'll get to hear it from a little bit different perspective. There is something called the primer that's put out by Jackie. The original one was put out in, I think, 2006, and then it's been updated 2 or 3 times with some outlines in much more detail provided here. In the past, I've had second years read this in addition to or in lieu of some of the Abbas chapters if they felt like they had a handle on it. Because we really do need some more detail for the boards than any of these textbooks are to provide. And this is where to get it. The latest edition, though, is getting out of date, so I'm not sure how much it's going to be now that these textbooks have been.
I like Dr. Belanti's book.
Dr.
The visuals, yes. I mean, the diagrams and the visuals are just great.
Yeah, yeah. Right, so that may be another one to go through. I guess we're going through January this year that you may not need anything extra to read, but I'll leave that to you. Announcement about the books. And I'm just the messenger. And this goes to the faculty too, but everyone in the hospital had their books funds canceled. So you guys still have your kind of book travel fund, but there's no separate book fund, and that's what we paid for the books out of before. This just came to everyone's attention. So probably purchasing these books, unfortunately, I'm sorry, it's probably going to get transferred to your fellow account. But it's not just you. The same thing has happened to us. So if that lessens the blow at all. And henceforth, that is unfortunately going to be. So next year, so you can negotiate your best price, we'll probably have you guys buy them on your own. Okay. So first years, as we go through, this can be very, very overwhelming, and you are very likely on several occasions to read through 3 straight pages and then think back and think you did not learn one thing. Don't worry about that. That is it for every single person who has ever done this. Trust me, you're picking up more than you think. It's a phenomenon. It's happened to everybody. You've gone through, you've learned absolutely nothing. You're picking up something. Try and see the forest through the trees. Try and just pick up the key big concepts. Don't try and sit there and memorize the details. So there'll be big tables as we go through with lots of words. Don't sit and memorize them. Kind of get a gist. Focus on the big picture and then focus on clinical relevance, especially as you see patients, how this all kind of comes together. David, however, you get to memorize the details this year. Review the key concepts, memorize some more, and then really try hard to apply the basic science in clinical disease. And then when you're done with that, memorize more details because you'll need them for the boards. The thing with immunology is it seems overly tested compared to clinical, in my opinion, on the test because it's easier to write questions. You have a definite answer for these things. So you need to know a lot of immunology for the board. If you wait until the last second, you will never pass the board. Start now studying for the board by just going through this class, and you will probably be in good shape if you read the textbook through 2 times in your 2 years here, attend most of the lectures, go over the Jeopardies over and over, you will likely be in very good shape. Okay, so expectations for me, I do a lecture about weekly, but the syllabus is up. I think I sent it to you. It's in the immunology folder, which is in the allergy folder on the W drive. And then afterwards, I'm just going to write a Jeopardy for each section with help from our colleagues on the East Coast this year. And then I'm going to be available for questions from anyone inside the institution or outside the institution. And my email address is there. Fellows, the most important thing you can do is just read prior to class. Even if you feel like you're not getting anywhere, just read it. Then read it again next year. And the second time through, you'll realize that you've learned something, and it'll start to all make sense. And then as you review it and memorize it, you'll realize you're in very good shape. Be here at 10. Please quit whatever you're doing and get here at 10. Try not to bring your computers and cell phones in text. It's just distracting for me. I get a little ADD in that way. And then if you get paged, try and handle it quickly and come back. All right, any questions about the format? Pretty straightforward. Okay. So this is my quick down and dirty review of the immune system. And then what we're going to do is cover each little part throughout the year. Okay, so right, what does the immune system do? Of course, the immune system fights infection, it fights cancers, it prevents autoimmune disease. But the immune reaction in a very simplistic format always starts the same way. Right, so you have a group of cells that are sentinels that kind of sit in the skin. They're immune cells. In one way, I usually point out that we kind of know these are sitting in the periphery or near where antigens can get into our bodies. Is that it's not something that we see when we check a CBC and actually look at the immune cells or the white blood cells in the blood. Right, so these are usually, or these are always the macrophage, the dendritic cell, and the mast cell. Okay. So if things get going, one of the first big players is the dendritic cell. That's what I'm going to cover first. So what the dendritic cell does, right, you might remember the Langerhans dendritic cell, the Verbeck granules. All the same thing. We'll phagocytose or eat or sample the antigen that's got in the environment it is. And what it's going to do is it's going to hop in the lymph, and it's going to travel to the nearest lymph node. Right? So now the lymph node is going to get the immune system started, and it does that by going to the T cell zone of the lymph node. And it's going to test many T cells, but eventually it's going to find a T cell that recognizes what it's displaying, or recognizes this antigen. So you're going to have a T cell specific for that antigen that can kick the immune system off. Does anybody remember what the antigen is being displayed on?
Like MAC.
Yep. MAC, exactly, right? So if that's a CD8 T cell, it's perfect, and if it's a CD4, it's a type 2. Perfect. In that is a protein. So that part of the antigen that it's sampled that's holding up there is a protein, that's necessarily a protein. That helps me remember the fact in food allergy that when we have a food allergic reaction, it's usually to a high protein food. Proteins are pretty easy to have immune reactions to compared to carbohydrates or lipids. I tell the residents when you're doing your boards, don't get tricked into thinking a banana caused the anaphylactic reaction. It's not that it can't. It doesn't usually. If you can't remember what food, you can't figure out food, will really hone in on those proteins. That's probably where the answer is going to be. Okay, so the dendritic cell is going to activate the T cell at this point. And what the T cell's first job is going to find a B cell that is also specific for the same antigen. So it's going to come to the B cell zone of the lymph node, where it's going to find a B cell. Now that B cell has known to kind of travel towards the T cell zone because little bits and pieces of that antigen have probably gone through the lymph and are filtering through the lymph node and have been caught in the web in the B cell zone. So it kind of gets activated and it moves. So now the T cell and B cell are activated. So if this interaction can't happen, if that T cell cannot activate that B cell for whatever reason, then you have a type of immune deficiency called, anybody guess?
Combined?
Yes, severe combined immune deficiency, SCID, exactly. Right, so severe combined immune deficiency, I'm not really going to talk too much about immune deficiencies, but very quickly, right, you don't have T cells at all, you T negative for practical purposes right now. And you may or may not have B cells, but it doesn't matter, because if those B cells can't get activated, it doesn't matter if they're there or not, and you have a combined immune deficiency. Both sides of the immune system can't work. The T side, nor the B side, cannot work. Severe combined immune deficiency. Okay. So now the B cell. So B cells activated, say it's there, you don't have SCID, what's it going to do? Well, it's going to eventually hop back into the blood vessel. It's going to go through the rest of the lymph, the rest of the duct, right back into the vessel. And typically it travels to the bone marrow where it hangs out and it secretes its product that does its work for it. The product, of course, is antibodies. And the first one it starts secreting is IgM, and then it goes to IgG or IgA or IgE. And depending on the type of infection and signals it's received throughout this process, it can fight infection effectively with these tools. If this part can't work, if that B cell cannot secrete these antibodies, then, well, you have probably an antibody deficiency. Right now, the antibody deficiencies are a little bit more complicated because there's we subtype them depending on your lab results and where the defect is. Right, so there is a possibility that you actually have no B cells at all, whereas, of course, you're not going to be able to make any immunoglobulins, and therefore, of course, you don't have any immunoglobulin response to vaccines because you have no immunoglobulins because you have no B cells. And if this is the case, then you probably have something like Bruton's AGAM, excellent AGAM. You have no B cells, no immunoglobulins, no response to vaccines, right? So this isn't the only form, okay, so there's some autosomal recessive forms of this too, but, you know, first one to think about is usually Bruton's, and we'll get to know the rest later. Okay, so sometimes, though, you have B cells, but for whatever reason, the B cells cannot make immunoglobulins, and therefore you have no immunoglobulin response to vaccines either. This is the case, we usually lump it into a category of diseases like common variable immune deficiency. So if B cells, immunoglobulins are not there or very low, and your response to vaccines is not there, very bad, we usually just lump you into common variable immune deficiency. And then, of course, if it's just IgA that's low, we call that selective IgA deficiency, and we don't really know if you have a lot more infections than everybody else or not. Maybe. Or you can have a problem where, well, sure, you can make IgM, that's the first thing made, but you actually have to essentially turn a switch on in the B cell to be able to switch from IgM to IgG or IgA or IgE, any one of those. If that switch can't be turned on, your body's just going to keep making lots and lots and lots and lots and lots of IgM. And it's not going to be able to make any of the other antibodies. So you have something called hyper IgM syndrome where you're relatively immune deficient because you're not making good high-yield antibody. Okay, and then there's a couple more situations. One could be that you have B cells, and sure enough, they're making immunoglobulins. But when you check your response to vaccines, particularly the carbohydrate vaccines like the Pneumovax, the adult pneumonia shot, you really don't have a good response to it. Clinically, you're acting immune deficient. Well, we call this specific antibody deficiency in general. May or may not treat with immunoglobulin replacement. Another situation that we see all the time where you have B cells, your immunoglobulin levels are very low, but it doesn't seem to matter that much because your response to vaccines is adequate. So although you don't have very good immunoglobulin levels, when you check all the different vaccines, well, you do have good levels of specific antibody. Well, what's happening here? Probably one of two things. If it's a baby, you may have something called transient hypogamous infancy. Or you may want to start looking for acquired secondary secondary immune deficiencies. Not primary. These others we discussed are primary immune deficiencies. Which could come from any of these things, probably most often medication, autoimmune disease. I feel like we see a lot of protein losing. Certainly here at this hospital we have cardiac surgery, protein losing a lot. Okay, so well, what do the T cells do? Well, it's going to be jumping back into the blood vessel as well. And it is going to get back to the site of infection where it's going to kind of act like the quarterback, and it's really going to start telling the other cells what to do by sending signals, and those signals are sent by something called cytokines. Don't get hung up on some of these terms. It's just learning a new language and getting used to them. So cytokines are basically just signals from one cell to another cell. A chemokine is a type of cytokine that tells a cell to go somewhere. So don't let that fool you. Cytokines and chemokines. So the T cell is going to send a message to the macrophage, which is a really, really good eater. It's going to phagocytose all the antigen. It's going to get revved up. It's possible this interaction can't happen, and you may have something called interferon gamma deficiency or IL-12 problem. But as a general rule of thumb, the macrophage is going to get revved up and get rid of that infection. Now, a macrophage only going to live so long, so if it's done its job and it actually dies itself, what type of cell is going to come in that's circulating the blood vessels and replace it?
Neutrophils.
To replace it. So if you are looking at your white blood cell count and you have your differential, so those are your options of immune cells that are circulating in the blood. Right, and we've already kind of talked about the lymphocytes, which would be T's and B's, and then, of course, NKs, and I'm not really going to talk about them today. But then what's left? Well, there's right, the granulocytes, neutrophils, basophils, eosinophils, and then there's one more that's not there.
Oh, mono.
The monocytes, right. So the monocytes are precursors to macrophages, so when you need more macrophages at a site, the monocytes will come in and take their place and fully mature into macrophages. Okay. Now, if the macrophages during the time of infection get overwhelmed, well, there's another really good phagocyte that can come in and get made very quickly and come in and help. And David's exactly right that is the neutrophil. Right? So they're pretty dumb. I call the flash mob of the immune system. They jump in, They eat, they die, they become pus. That's it. But they're really, really good at that, right? If you don't have neutrophils, you're probably going to die much more quickly than if you don't have lymphocytes. Right, just another type of phagocyte can be made very quickly. So what does that neutrophil actually have to do? What do any of these cells actually have to do to get to that site of infection? Well, we know that neutrophils are circulating in the blood. We know that because when we check CBC with differential, well, we see the neutrophils. We know exactly where they are. They're in the blood. And they have to get to the site of infection. Well, how do they do this? They actually come to, they go along the blood vessel, and then they stop, they slow down, and they stop. It's done by something called leptins and integrins, something we're going to learn about. When it does that, it adheres to the side of the wall, and it flips out to the site of infection. If this can happen, if it can't adhere to the side of the wall and get out, then you probably have something called leukocyte adhesion defect, right? So leukocyte adhesion defect means you've lost that adhesion molecule on the outside of the cell. Now this is a sticky point in immunology. So that adhesion molecule is called CD18. So in immunology, immunologists like to make things very, very difficult, or they like it their way. In order to have it their way, something doesn't have a name they like, they'll just give it a different name. There are many names for every single thing, and you need to know them all. There's no one standard that you can go by and get by on the test. You really need to know all the different names for all these things. So adhesion molecule is also called CD18. And we'll learn some more aliases as we go along. Right, okay. So cells have gotten to the site of infection. So once those phagocytes are there, what are they going to do? Well, right, they have to eat. And to eat, they bind the microbe, they bind the antigen, that microbe that's gotten in, and then they phagocytose it, right? So they got this membrane that zips around it and engulfs it into this phagosome. Now, the phagosome is going to bind something with something called the lysosome. It has a bunch of enzymes in it, and it's going to form a type of lysosome, and this is where the microbe is now going to die inside the phagocyte. Up to this point, it can still be alive inside the cell. So this is the point that actually mom is very good at. Right? When you get a cut, what does mom do? Mom pours hydrogen peroxide in your wound to kill the bacteria in it. Well, the macrophage actually, or the neutrophil, essentially does the same thing. So it takes oxygen and it makes it into reactive oxygen species that are like hydrogen peroxide, that are very good at killing these microbes. Now, you can't, your body is incapable of making oxygen into these reactive oxygen species. In other words, if you don't have phagocyte or NADPH oxidase, this mechanism that makes oxygen into reactive oxygen species, well, then you may have something called chronic or granulomatous disease, which should be diagnosed by actually looking with a flow cytometer to see if you have an oxidase first. There can be another problem with this, and that these lysosomes cannot move and bind with these phagosomes very well to make these phagolysosomes. If this is the case, the problem with lists, you may have partial albinism. You may have something called, any guesses? Chiari-Gashi syndrome. They actually would probably get diagnosed just by looking at a CBC and seeing these giant inclusion in the body. Right, okay. So that's kind of it. That's about, in very broad strokes, how the immune system works. However, if that first microbe that got in was not, say, a bacteria, and say it was a helminthic parasite that has a very thick skin, and does not get killed very well by reactive oxygen species, well, that's when the mast cell comes into play. And what that does is it keeps all allergens in business. But when it's working correctly, it should burst open very quickly because it has preformed mediators like histamine that are very good at chewing through the thick skin of these helminthic parasites. and it will dissolve them. Now, mast cells, what do you remember about mast cells? Well, the thing I like to try and remember about mast cells is where a lot of these other cells take many hours to get revved up and act. Mast cells, all the mediators it wants in that first line reaction are all pre-made. So the reaction happens very quickly, within 15 minutes. Nothing needs to be made. As soon as it's ready to work, bam, it secretes what it needs to. When we think of allergic reactions, mast cell-mediated reactions, very, very quick. Okay, now similar to the macrophage, if it needs help from something in the blood vessel, something that can be made very quickly, it's getting overwhelmed, well, it's going to call in the last cell from the blood that we had not talked about really, the eosinophils. The eosinophil is going to come in, and it's going to play the same role as the neutrophil does to the macrophage. I didn't really talk about basophils. Basophils are roughly blood equivalent of the cells. There's some differences, but I think that's all we need to know for right now. So, right, so when you look at a CBC with differential, what do you see? You see neutrophils, the granulocytes first, which are the neutrophils, neutral staining, the basophils, the blue basic staining, the eosinophils, that nice red acidic staining. And then, of course, the monocytes, and we figure out where they go, and then you see the lymphocytes, and lymphocytes are T's and B's and NKs, and we talked about T's and B's, and we will talk about NKs. So pretty much I just named the vast majority of the cells in the immune system. Now what gets taught is that some of these cells are going to be called different things as time goes on. Now, if I was a betting woman, most unusual cell names, if you can't readily place them, like a Kupffer cell, you know, it's part of the immune system, well, it's probably a macrophage. So it's not always going to be right. But if you look at a cell and you're like, whoa, this is not in the very basic list of immune cells that Ciaccio gives me, just guess macrophage. It's probably what it is. Macrophages get renamed in every organ system for whatever. I wish it wasn't so, but it is. Okay. That is about it. We have a few more minutes. Let me quickly talk about how we evaluate the immune system. Right? So our methods of evaluating the immune system aren't fantastic. I wish they were better. I wish there's a way, there's tests we could do to see how strong your immune system is. I know it's a very general term, but there's no way to do that. So we do some series of tests, right? We like to quantitate and qualitate things. We can never do one of those, just one of those. We have to look at both, try and make a determination how well your immune system works. So when we quantitate, what are ways we quantitate? Well, we would actually just physically count how many cells you have. And the way we often do that is to use a flow cytometer. So what's a flow cytometer do? A flow cytometer has a laser on it, and that laser is sent out and it hits the outside of the cell and then bounces back and talks to the computer. When it talks to the computer, it says yes or no. I did see what you asked me to look for on the outside of the cell, or I didn't see it. And you tell that laser before it goes out what you want it to see. So if you want to count up how many T cells you have, say you send 100 cells in front of that laser, and you say, tell me how many of those are T cells. Well, you'd ask that laser to look for something that you know is on T cells, only on T cells, no other cells. Well, what would that be? If I had to think about it, well, one that would make sense would be a T cell receptor is only on a T cell and not on any other cell. Okay, well, that's all well and good, and that's exactly what we do, but we're immunologists, so we don't call it a T cell receptor. What do we call it? We call it CD3. So you ask your flow cytometer to look for CD3, and then it would come back and say yes or no, and then we know how many white cells you have, and we know what percentage of those are T cells, because we've gotten a yes-no answer back from that laser. And we can multiply those two together, and we can see how many T cells about you have floating around in your blood. Right, same for B cells. So you ask the laser, oh, how many B cells do I have? Well, what's something that's on the outside of the B cell and not on any other cell? Well, a B cell receptor is on the outside of B cell, no other cell. Well, but we're immunologists, so okay, well, let's call it CD19, CD21, these are parts of the B cell receptor. And quantitate how many B cells you have for us. B cells, though, you can also quantitate in a special way. You can quantitate the product of B cells, too. We can't really quantitate cytokines so well. But B cells you can, so you can just get quantitative immunoglobulins. You can quantitate the number of IgG, IgM, and IgA, IgE in your blood. And that's very helpful. But then what we want to do is we want to qualitate and see how well these things work. So when we qualitate the immune system, well do our T cells and our B cells work. So one thing we can do is we can take something called a mitogen, which is a little protein that is found in a plant, usually exploited by a plant and plant defenses, but we then use it to put in a petri dish with a bunch of T cells. And a mitogen, if it sees a cell, should force it to go into mitosis. So mitogen forces mitosis. It should force it into dividing. Well, when that happens, that tells us something qualitatively about the T cell. So if a T cell is capable of dividing, if it's capable of responding to a mitogen, we like to see that. That tells us something qualitatively about the cell. Same for B cells. We'll just put with a mitogen. Let's see if it can divide. Alternatively, we can use something called an antigen, which, right, something, an infection you know you have had before. You should have a T cell and B cell that responds to it. Infections we know we have, well, that would be vaccines. So things like tetanus, you can use an antigen like tetanus and put it in a petri dish with T cells and B cells. And it should cause at least some of them to start dividing. They work well. And then we can qualitate immunoglobulins by looking and seeing if there's any that have been effectively made that is specific for something. Specific for an infection we know you had. Again, an infection we know you've had would be a vaccine. So you should have some immunoglobulins that are specific for the vaccines you had that qualitatively tells us something about your B cells. Quantitative quality, it's not all that always that perfect. So right, if we talk about the different parts of the immune system, in general we talk about the cellular immune system, which for whatever reason refers to the T cell side. And then there's the humoral immune system, which is the B cell side. The humors are actually the immunoglobulins. Before we knew what immunoglobulins were, we thought we were just transferring the humors from individuals to the so that name has stuck, the humoral immune system. All right, and then there's the phagocytes. And the phagocytes you have to test differently and individually. So, right, we do things like the oxidative burst to see if they've got. We look with a flow cytometer to see if there's an adhesion molecule on the outside of the cell. And I'll tell you if you have LAD. And any more, right? Well, we want a molecular, genetic molecular diagnosis, right? So we'd maybe do something like exome sequencing or genome sequencing, and actually, if we can't readily find what gene is defective, we may actually just sequence your whole genome and try and figure it out. So that may give us a better predictability about the nature of disease and maybe will guide us about how to treat it. All right, any questions?
Do you separate the workup into like tiers at all? I mean, I feel like a lot of our consults so far have been SCID, and so we're doing everything. I mean, if you have a team that just calls and says, hey, in general, we're concerned about immunodeficiency, do you have sort of like a, these are my basic initial steps, if those are concerning, then I proceed with this? And
yes, yes. You guys are seeing it very skewed. We've had a lot of SCID in the past couple months. Really an amazing amount. You're seeing skewed perception or version of what we do. So the first line is that you may just do history and physical, and that's it, right? So that's really going to send you, hopefully it's going to send you down one system. So, okay, well, T cell stuff probably going to be pretty easy or obvious. It's going to be cellular thrive. It's very, very young, unless it's acquired secondary HIV or something. But, you know, the infections are kind of, you know, are very characteristic infections, right, the atypical stuff. Or it may be humoral B cell, where we think of strep pneumo and strep pneumo again and strep pneumo again and strep pneumo again and strep pneumo again, because we really have a lot of trouble with strep pneumo. So ears and pneumonias and sinus infections and even UTIs. And not just the history you really want to decide where you're going to start sending tests. The history you really want is more than one organ system or something that tells you it's systemic, right? And we kind of intuitively know this, but if you see a baby that's had 7 ear infections in the past 8 months, well, we don't just jump to immune deficiency.
Right.
If we see an adult who's had 7 sinus infections in 8 months, we don't jump to immune deficiency. What do we jump to? We jump to anatomic. Isolated from one place, it was anatomic, right? So in a baby, well, maybe their eustachian tubes just aren't at a very good angle right now. We'll give them time. We'll put tubes in. An adult, well, were you a football player? Have you been in a car accident? Let's do a CT scan. Is there something obstructing, blocking that you can't clear your sinuses? But if it's in 2 spots, like if you got admitted for a pneumonia and had ear infections, well, that gives us a sense, well, maybe this isn't anatomic. There's no anatomic, exactly, easy anatomic explanation for that going on. That's systemic, and systemic makes you think immune deficiency. Right, so then, and phagocytic stuff, you think more like gingivitis and ulcerations, and it's a little tricky because in little kids, you may see just repeated infections, but you may see like a super high neutrophil count to make you think of LAD. You're making the neutrophils and they don't come out, right? Ideally, you could at least compartmentalize one of those as your high suspicion. Or the first thing you say is, I've had this conversation, and yes, I believe there's a chance you have immune deficiency, or no, you do not have immune deficiency. And sometimes it's just reassurance, right? So then, okay, yes. And yes, if I was a betting woman, I think it is T cell or B cell or phagocyte. The vast majority of the time, what you see is, okay, maybe you have an immune deficiency. It usually points you to the B cell. They're older, and it's a lot of sinus infections and lots of ear infections and that type of stuff. So then what I usually start with is just quantitative immunoglobulins in response to a couple of vaccines. So maybe tetanus and Hib and diphtheria, or sometimes you get lucky and they've already had their rubella levels checked for some other reason. Hepatitis, really just need a couple. You really need more than one because, well, some percent of the population, all comers aren't going to respond to every vaccine series. Tetanus we check all the time because it's really good. So upwards, I think it's actually over 95% of the population responds to tetanus. So you probably are going to see a tetanus response, but you don't want to label someone immune deficient if it's not there, because there's still going to be those people who haven't responded well for whatever reason. We're just starting to understand that maybe you were sick when you got the vaccine and it didn't work as well. Maybe you were got a bunch of Tylenol when you got your vaccine and you didn't respond well. Okay, but everyone gets a chance to fail a vaccine. So we just check another one, and then 2 types, protein and carbohydrate. So probably something to look at strep pneumo titers. So the most basic workup that I do a lot would be quantitative immunoglobulins, vaccine titers. And especially if my suspicion's low, I would end it there and say, I think you're good to go. Now that's different than if you're getting called to the hospital for someone. It's usually more expensive because there's a reason they're in. Probably ID's already on board. If ID's on board and thinks it's immune deficiency, then I take it very seriously because they usually know. Or they may say, here's the thing, we have this chromiobacterium violation, and that is, you are only infected in that if you have CGD, right? So you may know from bacteria, and there are lists you can find on like CIS website. If they name the bacteria, then the only thing you need to get is an oxidative burst. If someone's infection after infection and their white count is through the roof, now leukocyte adhesion defects we think about. Your body's making those leukocytes, but those leukocytes or white blood cells just can't get to the site of infections. So you just keep making more and more and more. So yes, it's a tiered approach. And then antibody stuff gets even more complicated because sometimes I send antibody and vaccines, and then hindsight, I wish I didn't, because deep down in my heart, I really didn't believe they're immune deficient. So then I put them into a category. Well, now do I think they're immune deficient? Or now seeing what I see in front of me, do I still think they're immune deficient? And I kind of categorize them. Then I start, everyone starts playing this game with the pneumococcal antibodies. How high are they, and how much have they increased? And do you have 7 out of 14, or 9 out of 14, or 2 out of 14? And ultimately, at that point, I don't care, because ultimately, at that point, I have a really strong gut feeling whether they're immune deficient or not. So I use that as either A, evidence to give to the insurance company that I need to start immunoglobulin replacement, or evidence I give back, and I may take the same numbers depending on the clinical situation, and in one hand say, no, I don't think you're immune deficient. On the other hand say, yes, you're immune deficient. There's a lot of clinical direction at that point. We all get stuck in that scenario, unfortunately, and there's no great way out of it, but you should know it's usually okay to just watch and wait, especially if you really don't think you're immune deficient. Their antibodies are a little bit off. You know, you're probably not in a hurry to start immunoglobulin. You probably can just wait and see if things go. So those are some tried and true rules that I would buy. Yeah. But it's very complicated. There's no quick answer. You probably, well, actually, I have to say you probably won't see as much skid, but now that we're going to get to the newborn screen, you probably
for sure.
Yeah, for next year, even this year. So you'll be experts, so that'll be great.
How much are these tests, Dr. Burns? Like, do you know, like, how much does just flow, like a flow cost?
Depends on where you go to get them.
Okay.
So a lot of times, the last prices I saw out of Viracor for simple flow, they could turn one out for like $300 or so.
Okay.
If you go for a big workup at Children's, I think they're like $4,000 or $5,000.
I think we do like 300 or 400 per.
Yeah, per recordable.
Marker.
Yeah, per recordable marker, and they're like 10 markers. So
if you do 3, 4, 8, 19, 15, 56, you know, take that.
Wow.
5 times 300 or 400. So you're looking
at. And then with the extended the EIDP and then the naive in memory, like we've been
right.
I've had to. That needs to
right. But that's another good one. If your suspicion's lower, you may just want to get B's and T's.
Yeah.
Yeah.
So you know, it little depends on where you go and how much you order. Really.
The state of California got a deal where they got flow to follow up the newborn screen, I want to say for $9.
Oh, wow.
Yeah, I mean, you know, if
but
you
clearly it's not a ton.
Yeah, well, if you run a lot of them,
Yeah.
you know, the cost is your machine and your person to run it and do the prep.
Yeah.
If you run a lot of them during the day and you keep that person busy and you keep that machine occupied for 24 hours a day, you get them done a lot cheaper than if you've got one person who does maybe one of these a day. You know, so.
True.
And then, and the other thing you have to consider is how much follow-up are you getting? You know, are you just going to get the number back? Are you going to get the number back and a pathologist you can call to explain what this number means or tell you how the test was done and that kind of thing? So, you know, you kind of get what you pay for a lot of times.
Right, because you're not quite sure what.
Right.
Yeah. Yeah. Is antigen and myogen stem expensive too?
The prices have come down considerably. We don't do those here anymore. We can get a panel by separate mechanism from Mayo for like $400, I think. And we used to, those things used to cost $700 to $800. So.
Kind of a moot point when they're inpatient, because I think all those get bundled into
I'm sure.
just one general.
Yeah.
I know genetics likes to send all the genetic tests as an inpatient.
Yeah,
they get bundled.
And it won't be long here before you're going to have to justify extensively any genetic test you send out.
Yeah.
Because I know that's on the pathology to-do list this year, is that you will have to justify any genetic test that goes out of here.
Yeah.
Because I mean, they're spending millions of dollars.
Yeah, I'm sure.
On genetic testing, and so they at least would like it done properly in a logical order, rather than just, we'll send this one and when it comes back it's negative, we'll send the next one, and when it comes back it's negative, we'll send the next one. Because the argument is, if you're going to send 8 tests, why not just throw it in and do the next gen sequencing, or do a big screening test, do the flow cytometry, do the cytogenetics, narrow it down and then go from there. So,
That's true.
yeah. And those fields are changing all the time.
Just like my consult last week, they just ordered, the farming team just went ahead and ordered a fish. The lady for any reason to order it. But they were just like, let's just roll this out. Yeah. And,
Yeah.
Okay.
So next week, comparisons 2, 3, 4 we'll go over.
thank you.
Thank you.
So you want to turn off recording? Oh, you did. Okay. I see them on my desktop, so I will load them in that.
Oh, I didn't